Hey everybody, Bob Long, your anatomy and physiology professor at Del Mar College. For those of you that are enrolled in my class, this is for part 1 A&P. This is muscle lecture number 3, the third in a series of lectures. Again, I'm trying to keep the muscle lectures, or all of my lectures, to about 10 to 15 minutes as much as possible, under 20 minutes, simply because of the difficulty of managing files and uploading stuff. And also for you to be able to keep up with the worksheets that I've printed on my Canvas page or in the note set. So anyway, as we discussed last time, we went over the structure of a muscle, from the muscle to the fascicles to the muscle cells. Actually, I didn't draw this quite correctly, but if this were a fascicle, this would be a muscle cell. This would be a myofibril. So what we're going to start looking at is the structure of the myofibrils and the myofilaments, actin and myosin, as we discussed them in the last video. So what I'm going to do is I'm going to cut out a section, essentially this section right here of this myofibril. I'm essentially going to cut out a structure called a sarcomere, and we're going to draw it out and learn it together. If you're trying to follow along in your notes, then the structure of the sarcomere is going to be on the bottom of page 53, and there are some definitions at the top of the page that you should fill in if you have your notes set. All right? So look, if I were to take this structure, the easiest way to do this is over near the edge of your page, wherever you're drawing. Draw a zigzag-shaped line like this that essentially has 4 points. We're going to have some little red lines coming off in this direction, which would be these little proteins over here. And then I'm going to have some red lines coming off, and I'm going to go over a specific distance, and I'm going to make all of these lines the exact same distance. In between each one of those, I'm going to color code this so it matches our textbook, but overlapping just a little bit, not all the way over, I'm going to draw 4 lines that run a long ways across the board here. Now, on the other end, overlapping the same amount, or roughly the same amount, I'm going to draw 4 more red lines, like this, and then I'm going to connect them like so. And then sticking off here would be these proteins on the other side. Now running right down the middle is going to be a thick band like this. Okay, I'm going to fill this in. Now what we are seeing is sort of a flat structure, 2-dimensionally, but in 3 dimensions, this would actually go around sort of in a ring like this and go on the other side, so that we're looking at a sort of a rounded tubular structure of protein. This structure, these zigzag-shaped lines, get a specific name. So each one of those zigzag-shaped lines is referred to as a Z-line. So at either end, I have a Z-line. And the area that covers from Z-line to Z-line all the way across is known as a sarcomere. Sarcomeres are repeating subunits. There's a sarcomere here, and they would go all the way down the length of these myofibrils. They would continue to the other end as well, in every myofibril. It's a very unique and highly ordered arrangement of these proteins. Now, the little red lines represent the protein called the thin filament, and sometimes I use the term actin interchangeably. I know that's not 100% accurate, but this is our actin filament or thin filament, and the little purple lines are going to represent the protein myosin, or the myosin filament or thick filament. And the line right down the middle of the sarcomere is called the M line. It's going to get a little bit messy, but you got to learn to practice to draw this out so that you can understand, because it will help us understand how muscles contract when we get to the next lecture. Now, a sarcomere extends from Z-line to Z-line, and then we'd have another sarcomere from Z-line to Z-line, and so on. Within the sarcomere, there are subregions. One of the areas of the sarcomere, and I'm going to try to use some different colors here, that is comprised of the area that has only actin filaments, and quite honestly, if I were to look at it, it would go up here from here to here on either side of the Z-line, including the actin on either end. This area of a sarcomere that has actin only is known as an I-band. And if that's an I-band, then this area over here that has actin only is an I-band. And if you're trying to define this, a sarcomere goes from Z-line to Z-line. A Z-line is sort of a three-dimensional plate of proteins to which the actin filaments or thin filaments are anchored. So these are attached here. Imagine if I could cut this and turn it sideways, I would have a big ring of protein like this, and I would have these little sticks sticking off in two directions, sort of three-dimensionally. So this is a solid plate, almost as if I stuck a bunch of spears in the wall and they were sticking out at you. The wall would be the Z-line, the spears would be the actin filaments. The same thing happens here at the M-line. The M-line is a solid plate of protein to which myosin filaments are attached. And the area in the middle of the two I-bands that goes from the end of the myosin filaments to the end of the myosin filaments, all of this area right here is referred to as an A-band. Now the terms I and A come from, back in the, I believe, the 20s or so, 20s or 30s, scientists figured out how to make a bunch of light waves travel in the same direction instead of spreading out. And when light was traveling in all the same direction, they called it isotropic light. When they shine isotropic light on different things, sometimes they see things that you can't see with what we call daylight or regular light that's being scattered in a number of directions. Well, they started looking at everything they could under the sun with isotropic light and seeing what happens. When they were studying skeletal muscle tissue and shined isotropic light across it, what they realized is that on the wall they would see a light band where the isotropic light was blowing right through and showed a light spot, and then the thick filaments, the light would hit it and scatter and it would create a shadow. So they called that anisotropic light. And then they had another band of isotropic light. And when you look at skeletal muscle under a microscope, as some of y'all have probably done by now, you notice the dark bands and the light band and a dark band and a light band and a dark band. The dark bands are the A bands, the light bands are the I bands. And it's because of these thick filaments scattering light, creating the shadow, and the light blowing right through where the thin filaments are. Anyway, that's where the names came from. So one sarcomere is divided into three bands: I-band, A-band, I-band. Each A-band is divided into smaller regions, and we refer to these as zones. So now this picture has gotten a little bit messy, and I really don't want to erase anything, but I'm going to have to. So I'm going to erase this up here, but I think you see the idea of how it all fits together. So when I erase all of this up here, within my sarcomere, if I look at my sarcomere, and I'm going to rewrite some of this, sorry, this isn't working out exactly as I had hoped, but that's okay, we're going to make it work. So when I look within my sarcomere here, there are different areas of the sarcomere. And again, the sarcomere goes from Z-line to Z-line over here. This area of the sarcomere that has both actin and myosin overlapping on both sides here. If I look in this region, I see actin and myosin. In the I-band, I only see actin. In this area, I see actin and myosin overlapping. So I'm going to put a little asterisk here, and that stands for an area called the zone of overlap. A zone of overlap has actin and myosin overlapping each other, which creates this final area in between the two zones of overlap where I see myosin only and, of course, the M line. And this area that goes from this zone of overlap to this zone of overlap, the area in between, and I'll put a blue asterisk there, that is called the H zone, or some books call it the H band. So I learned it sort of this way. A sarcomere has three bands: I-band, A-band, I-band. The A-band is divided into three zones: zone of overlap, H zone, and a zone of overlap. The I-band within a sarcomere is the area or the region within a sarcomere where we see only the actin filaments. The Z line is where the actin filaments are anchored. The area of a sarcomere that we call the H zone is the area where I see only the myosin filaments and no actin. And it also includes the M line in the middle. The zones of overlap are the region of the sarcomere that contain actin and myosin overlapping. And then if I put the zones of overlap plus the H zone together, I get the A band. So you can write out the definitions of these things this way. I'm going to try to follow the order that's in my note set. So a sarcomere is a region that extends from one Z line to the next, and it is the contractile or functional unit of the muscle. The Z line is a three-dimensional plate of proteins to which the actin filaments or thin filaments are anchored. The M line is a three-dimensional plate of protein to which the myosin or thick filaments are anchored. The I band is the region of the sarcomere that contains only actin filaments. The A band consists of a zone of overlap plus an H zone plus a zone of overlap. The H zone has myosin only. The zone of overlap has actin and myosin filaments overlapping each other. Those are sort of the verbal definitions or descriptions of a sarcomere. Now in the sliding filament theory, which I mentioned in the last video, what's going to happen is, in a minute, we're going to do another video where we look at the actual molecular structure of actin and myosin. And what we're going to see is that the myosin filaments have these little tiny heads. And the little heads of the myosin filaments will stick out like this, and they're stacked almost like little golf clubs. And because there's so many of them laying over each other, it makes it very thick. And they'll stick off in a number of directions. And all these little myosin molecules are stacked like this. And given the opportunity, when we understand the three-dimensional structure, these little myosin heads can extend up and grab on or bind to and attach to the actin filaments. And when they do this in both directions, they will literally grab onto the actin filaments like my arm's grabbing a rope and pull. And when this happens on both sides, and some of them are attached while others are still reaching up, when they grab and pull, they'll pull the two Z lines towards the M line, causing a sarcomere to contract. And if you go back to the last video, and I look at the length of a myofibril, if each sarcomere contracts just a little, it will pull the ends of the myofibrils towards each other, shortening them. Since the myofibrils are attached to the ends of the inside of the muscle cell, it will contract the muscle cell. Since the muscle cell's endomysium is attached to the end of the fascicle and tied into the peri and epimysium of the muscle, when the sarcomeres contract and the filaments slide past each other like this, then the myofibril contracts, the muscle cell contracts, the fascicle contracts, and the overall muscle contracts. So muscle contraction really begins with the interaction of these small proteins, actin and myosin, or the thin and thick filament. So now that we know the structure of a sarcomere, we'll talk about the molecular structure of actin and myosin in the next video, and then we'll talk about how do muscles actually contract. It's a series of steps. And then finally, we'll talk about muscle performance. And you'll take quizzes after each one of these, those of you that are in my class. The rest of you, I hope that you gleaned some information from this and it was helpful. So hope you had as much fun as I did. Hope you learned something. Thanks.